It's fancy, but I need to make sure I get everything in today. All right, so we're doing muscles. Okay, yeah, muscles and the muscle tissue. So we're looking at macroscopic and microscopic anatomy of muscles here. Okay. So whenever we discuss the 4 tissue types, we said that there was epithelial tissue, connective tissue, muscular tissue, and nervous tissue. This entire chapter is over the muscular tissue. And I also mentioned when we did tissue that there were 3 types of muscular tissue. Skeletal, that's attached to bone, smooth, that's on your viscera, and cardiac, which is in your heart, thank goodness. Yes. So 3 types of muscular tissue. The way this chapter is structured, I think there's like 120-something slides. 80 of them are skeletal muscle. Then we have smooth, then we have cardiac, and it's over. So we're going to spend the majority of this chapter on skeletal muscle. Okay? So I already mentioned that. Your terms that you're looking for that indicate to you that you're dealing with muscle. Myo, mes, or sarco. And this is not sacro, it's sarco with the R before the C. So sarco, sarcolemma, sarcoplasmic reticulum, all of it, sarco is muscles. Okay? Skeletal muscle is muscle that's attached to bone. When we talk about anything macroscopically and then we go into it microscopically, we talk about its cells. There is no skeletal muscle cell. We call skeletal muscle cells muscle fibers, and I'll explain this as we get to them. So muscle fibers are skeletal muscle cells. You do not see the same in smooth or cardiac, okay, as far as the terminology is considered. Skeletal muscles are striated, which hopefully if you worked through some of those labs, you heard that term or you read that term striated, you looked at that tissue, you saw striations, which means that it's striped. All skeletal muscle is voluntary, which means it's under conscious control. If you are not telling it to do it, then it's not doing it. They contract rapidly. They're super powerful, most of them, depending on their size, and we're going to discuss that today. But because they contract easily and they're pretty strong, they tire very easily. So there's quick fatigue. It requires nervous system stimulation. If your nervous system is not functioning, your skeletal muscles are not. So when you have people who are paralyzed, it's not their smooth muscle, it's not their cardiac muscle, it's their skeletal muscle. They're paralyzed because the nerve that controls them is damaged. Cardiac muscle is only in the heart. It is also striated, which means that it's striped. Okay? It contracts without the nervous system stimulation. So this is when you have a patient who's brain dead, but their heart's still going. They don't need their brain to tell their heart to beat. Your heart, in 2402, we'll discuss, has its own little nervous system, and it goes on its own. So it doesn't need the brain to tell it what to do. And thank goodness for that, because you and I would probably be dead if we had to remind ourselves for our heart to beat. Okay, so that's completely involuntary. And again, I covered more in detail in 2402. Smooth muscle is found in your viscera, so it talks about the walls of your organs, or your viscera. It is not striated, which is why it's called smooth. Okay, it's not striped, so it's called smooth. It also contracts without nervous system stimulation, meaning if you're brain dead, your organs can still go on without it. Okay? Your brain doesn't have to tell you to digest. You have a little nervous system, it's called the enteric nervous system, that controls your digestive patterns, whether your brain is working or not. Okay? So it is also considered involuntary. The only one that would be affected by the central nervous system would be skeletal. All right, this I feel like is a priceless diagram, and depending on which textbook you have, it may or may not be present. So what this gives you is the 3 types of muscle, and it kind of gives you a description. It shows you what it looks like under the microscope. Here are those striations for those of you who looked at this already, and you worked through that part of the lab. This should look familiar. This is skeletal muscle, striated. It's multinucleated. Okay, so we don't call them skeletal muscle cells. This is a skeletal muscle fiber. This is cardiac tissue. I know this is cardiac tissue because it's striated. I can see it, but these red lines kind of give it away. This is what we call an intercalated disc. If you worked through that part of the lab, you saw that as well. These are uninucleated. So an individual heart muscle cell is called a cardiomyocyte. So the myo tells me muscle, cardio tells me it's in the heart. Sight tells me it's mature. This is smooth muscle. Your tissue is not green. It's been stained for this purpose, but it is smooth as opposed to these two, which are striated. These are also uninucleated, meaning one nucleus per cell. Okay, so we have smooth muscle, not striated, involuntary, cardiac muscle, striated with intercalated discs, uninucleated, both of them, involuntary, both of them. Then we have skeletal muscle fibers, which are striated, multinucleated, and completely voluntary. Here, it's showing you what connective tissue is introduced or present in that specific muscle cell. The one you'll really only need to focus on is skeletal muscle, and we'll spend time on that in just a moment. Here it is at the functional unit level. This is a sarcomere. This is the contractile unit of a muscle fiber. You only find these sarcomeres in the skeletal and cardiac muscle, because that's what gives it its striations, its striping. It's clearly not present in the smooth muscle. This takes it further, and by the end of this chapter and / into our nervous chapter, this information will be covered. But this is showing you that each skeletal muscle fiber has its own yellow on it. Do you remember what yellow indicates we're talking about? Nerve. Every skeletal muscle has a nerve that controls it. So your brain tells it what to do. So that's showing you that it is innervated there. We don't see that in the cardiac tissue because the central nervous system does not control that. Okay? Here in smooth muscle, we see yellow, but it's spread out, and it's not 1-to-1 ratio here. And even though it does have its own nervous system, this is still coming from the central nervous system. It's part of your fight or flight. So, for example, if you are digesting and you're not telling yourself to digest, because of course you would forget to do that as well, but all of a sudden you get scared. Your brain sends a muscle to your digestive system and says, shut down right now. You are no longer working because all the ATP you're using, I need to use in my heart and lungs. Okay, in my skeletal muscle. So anytime you get scared, your brain will shut down your digestion. Anytime you start working out, digestion is shut down. Okay? And then here it's showing you how it is specifically innervated, and we'll get into that, and I will cover that more in detail when we go to the nervous chapter. Here is the level, and this is still that one diagram that I was showing you, just sections of it, all divided up. Here is showing you the actin and myosin, the troponin and tropomyosin, which we'll talk about today, and you'll see all of that today when we go through our sliding filament theory. And then when we talk about the speed of a muscle contraction, for your skeletal muscles, they can contract at different speeds. The smaller the muscle, the faster it will contract. The larger the muscle, the slower it contracts. So that just makes sense, okay? The smaller the muscle, the lower the load, so the less it can do. The larger the muscle, the larger the load. When you have cardiac and smooth muscle, we don't try to speed those contractions up. We want them to be meaningful and rhythmic. Okay, so it's giving you that there. Okay, special characteristics of muscle tissue. Muscle tissue has to be excitable, which means it has to respond to stimuli. That's what excitability means. So something tells it to do something and it does it. It needs to be able to contract. So muscles only contract. When they relax, it's the contraction being let go. Okay, so they don't force a relaxation. It's that the contraction has stopped. Extensibility. Muscles need to be able to stretch, but they also need to be able to recoil. There is a percentage of muscle fiber overlap, so you can stretch your muscles a certain extent, and as long as those fibers are able to grab onto each other, that muscle will still contract. If you stretch that muscle too far, you have torn it, because now these two can't grab onto each other, so that muscle won't flex. So we'll talk about that. Your muscles do need to be able to stretch, but they can only stretch so far without being damaged. And elasticity, the ability to recoil, which is finishing the extensibility aspect. 4 functions for the skeletal muscles, or muscle function in general. It needs to be able to move you. So skeletal muscle moves based on your bones, but smooth muscle moves fluids throughout your body. Your skeletal muscle also helps with moving fluids through a milking action, but we covered that in 2402. Maintaining your body posture and position. muscles. Skeletal muscle keeps you sitting up and doing things that you should be doing. Stabilizing joints. We talked about that when we talked about the joint chapter, how it was covered in muscle in order to strengthen it. It allows for heat generation. This is specifically talking about skeletal muscle and the individual muscles that are attached to each of your individual hairs. The erector pili muscle. That's a skeletal muscle. Okay, additional functions. Continues to protect organs, forms valves, and again that's going to be in blood vessels, as well as in the heart. Controls the pupil, so your iris right behind that is a circular muscle that opens, allows for your pupil to dilate or constrict, and also causes goosebumps, which is the erector pili muscle. That's plural, erector pili muscles. Okay? Every single muscle, when we're talking about skeletal, and it's telling you at the top we're referencing skeletal, every single muscle has one artery, one vein, sometimes they can have a few more, and one nerve. So one artery, one to a couple veins, and one nerve. Every skeletal muscle. So it has its own blood supply, and it has its own nervous innervation. Okay? Blood supply is important so that oxygen can get to our muscles. If oxygen cannot get to our muscles will fatigue. And they'll produce what we call lactic acid, which will build up and cause muscle cramping. So we need a blood supply because we need oxygen and nutrients. We need the nerve so we can tell it what to do. The vein is carrying back all the wastes to get rid of whatever waste were produced during the use or the contraction of that muscle. Okay? Yes. As far as the anatomy of the muscle, and I'm going to use the diagram on the next one, and I'm going to hope that it kind of works out. But here is me kind of showing you the association of these. Here is your large muscle. Your large muscle is covered by the epimysium. The epimysium is whenever you, and I'm going to go to a chicken bone again, but whenever you're eating chicken on the bone and you can see the tendon where it's attached, okay, when you pull that tendon away and there's like a layer that comes along with it that's not the actual meat, but it's on the outside, that's the epimysium. Every single one of our large skeletal muscles has that. It's what actually turns into our tendon. So that layer of connective tissue surrounds that whole muscle and it turns into our tendon. So here is our gross muscle. Every muscle is made up of small segments called fascicles. So fascicles are right here. Here's a muscle. Here's fascicle. I think I spelled that wrong, but whatever. And the fascicles are covered by the perimysium. Each fascicle is made up of multiple muscle fibers. And remember, because we're talking about skeletal, muscle fibers is technically the cell for the skeletal muscle. So this is a fiber. And there's a diagram on the next slide. I'm just trying to organize the thoughts. And each fiber is encased by the endomysium. Okay, if I was coming at this without knowing this material, but I had the ability to dissect these words, there's a lot I can tell you here. Just knowing the terms endo and epi. Epi tells me it's on the outside or the top. Endo tells me it's on the inside. Mes. Mes is a prefix or a root that tells me I'm dealing with muscle. So you're trying to put these terms together so that you can conjugate to some extent what we're talking about. Okay? And it's going to kind of show you that as we look at this diagram here. So here, first of all, where's this muscle located? How do you know? There's the femur right there. So you start, and anything I want you to do your best to gain as much information from that as possible. So this is my femur. I can see that by the structure of that bone there. It's not necessarily labeled, but I can tell that. Right here, this is the whole muscle together. And it's covered by this casing called the epimysium, and that epimysium will stretch and attach that muscle to the bone. It'll create my tendon. This whole muscle is made up of these things right here, and each of these things here is called a fascicle. So here it's kind of shot out one of those fascicles so you can see it. The fascicle is surrounded by this connective tissue here called the paramysium. Okay? Each fascicle is made up of units called muscle fibers. This is the cellular level of a skeletal muscle. This is the cellular level of a skeletal muscle. So here's a muscle fiber, and that muscle fiber is encased by the endomysium, which is the connective tissue there. So epi, peri, endomysium. Questions on that terminology? Yes, ma'am.
The fascicle and the muscle fibers technically the same thing?
No. A fascicle is a bunch of muscle fibers put together.
Put together, okay.
Yes, ma'am. Good question. Additional questions? Okay. So, this slide is actually taken from your bone chapter, and I said this is kind of an off thing, but I'm introducing these two terms. The term is origin and insertion. The terms, not term. Origin means where that muscle attaches to the immovable bone. Insertion is where that muscle attaches to the bone it will move, or what we call the movable bone. The example I gave you when we were in the bone chapter, whatever chapter that was the biceps brachii. The biceps brachii originates on my humerus. It inserts my forearm, ulnar radius. So when I flex my biceps, it does not move its origin point. It does not move my humerus at all. But where it inserts is what it moves. Okay, so it inserts on my forearm, ulnar radius. So when where it inserts, when I flex that muscle, it moves my forearm. Attachments for muscle can be direct and indirect. Direct means that muscle is right up against that bone and there's no really long tendon. Indirect means that there's a long tendon / attachment where it may fuse together. Okay, so direct attachment means it's already fused to the periosteum. And to remind you, the periosteum is that connective tissue on the outside of the bone that has blood vessels that serve the outside of the bone. Indirect means that it has like a tendon. The term aponeuroses is a tendon sheet. So you have an aponeuroses right here, like you don't have an individual small rope tendon here. You have a sheet of tendon here that's attaching this muscle and this muscle. Okay, so an aponeuroses is a sheet of tendon. So here again, showing you the whole muscle together with the epimysium, and then that epimysium coming together to form the tendon. Okay, the epimysium covers that whole muscle. That whole muscle is made up of segments called fascicles. The fascicle is surrounded by the paramysium. Then the fascicles are made up of multiple muscle fibers, so it's kind of shooting that out, and that muscle fiber is encased in a connective tissue called the endomysium.
When you build muscle, do you make more fascicles, or do the fascicles grow?
Great question. Fascicles grow in size. The muscle fiber itself grows in size. We call it muscle hypertrophy, which we will get to today. So you don't actually create more muscle fibers unless it's an extreme condition where, and there, you do have the ability, but as they're mature, they become what we call amitotic, which means they no longer divide and create more. So great question. It's that they get really big. The muscle fibers get big, causing the fascicles to get bigger, causing the whole muscle to appear larger as well. Okay, here is the muscle fiber encased in the endomysium, but get ready for this, because I wish I could just say, oh, there's just 3 layers that you need to be familiar with. No, there's not. You now need to know that each muscle fiber is made up of smaller segments called myofibrils. So each muscle fiber is made up of smaller segments called myofibrils, or myofibril. And I would like to say it stops there. It's anatomy. Each myofibril or myofibril is composed of segments called sarcomeres. So it's not an extension of the myofibril or fibril, it is within that. These are sarcomeres.
They're within.
The myofibril. So here, like this right here, is one, and this isn't a really high resolution picture. We'll see more in just a moment. But this whole thing would be this. So this is just this is that up close, and right here it's dividing it into sarcomeres. When a muscle contracts, you and I think it's like large scale, the whole muscle contracting. In order for a muscle to contract, this has to contract. So the sarcomere contracts, and in fact the definition of a sarcomere is the contractile unit of a muscle. The sarcomere is the contractile unit of a muscle. So the way it works is the sarcomere contracts, so the myofibril shortens. The myofibril shorten, so the muscle fiber shortens. The muscle fiber shortens, so the fascicle shortens. And because the fascicle shortens, the whole muscle shortens. Like it's that cascading reaction. Okay, so in order for your muscle to contract, it starts right here. And that's what we're going to focus on today for the sliding filament theory, is the contraction at the sarcomere level of the sarcomere. Questions right now? Go ahead.
The fiber is the cell, right?
That fiber is a skeletal muscle cell, correct. Yes. A muscle fiber is a skeletal muscle cell. You will not hear muscle fiber when we talk about smooth muscle. You will not hear muscle fiber when we talk about cardiac muscle. Okay? Yes, good questions. All right, so myofibrils, which are this smaller unit here, are made up of small segments called sarcomeres. Sarcomeres, as I mentioned just a moment ago, are the contractile units of a muscle. They are contractile units of a muscle. We have to go smaller than sarcomeres now. This is where you start hitting your head on the wall. You're like, why? Why? It's because when you give a patient medication, guess what level it impacts? The sarcomere level. It doesn't do the whole muscle. It goes to sarcomeres. And we're going to talk about troponin and calcium and all of that. Get ready. Hold on tight. Here we go. Striations, I've already mentioned what those are. So here it is up close. Here it is up close. In this diagram right here, I have a muscle fiber. A muscle fiber is composed of segments called myofibrils. Within this muscle fiber characteristics I can point out, it is encased in the endomysium. Okay, do you see how that endomysium all of a sudden becomes called the sarcolemma? Sarco tells me muscle. Sarcolemma is the cell membrane of a muscle cell. So why didn't we just call it the cell membrane of a muscle cell? Because we're doctors. And when you hear the term sarcolemma, you're supposed to understand that we're talking about the cell membrane of a muscle cell, which also ends up being it's continuous with the endomysium. Okay? As we get here, when we're looking at this whole fiber here, you can see these dark and light stripes. This is what we reference as the striations. Okay? And these striations are manifested as a result of the organization of the myofibril. I'm going to teach you what A bands and I bands are in just a moment, but where it's dark is what we call the A band. Where it is light is where we call it the I band, and I'm going to explain why all of that is the way that it is. You can also see on this diagrammatic representation that there are multiple nuclei present, because a myofibril many myofibrils compose a muscle fiber, and a muscle fiber is multi-nucleated. Skeletal muscle fiber. Okay? So you can see all of that here. I think that's all I would pull from that. Also look at mitochondria. The level of mitochondria in your skeletal muscle should exceed all of your other organs, except for your heart. Unless, if you are a really weak, physically weak person, you have more mitochondria in your heart. But if you work out a lot and you're very active, you'll probably have more mitochondria in your skeletal muscle. Okay? Here kind of gives you some terms. And I want to kind of discuss these terms. First of all, it says microscopic anatomy of a skeletal muscle. So again, we're still in skeletal muscle, and we will be for a while. I told you at the beginning. The muscle fiber itself is long and cylindrical. It has multiple nuclei. It is covered by the sarcolemma. The sarcolemma is the plasma membrane for the skeletal muscle fiber. Again, I said skeletal muscle fiber. Sarcoplasm. Sarco tells you muscle. Sarcoplasm sounds a whole lot like cytoplasm. But why call it sarcoplasm instead? It tells me specifically I'm talking about the cytoplasm of my muscles. Once you can get these terms, everything becomes so much easier. So sarcoplasm is the cytoplasm of a muscle fiber. Here we have terms. Glycosomes. Can you tell me what you think glyco sounds like? Sugar. So this is sugar storage, glycosomes. My muscles need glucose because glucose allows for ATP to be produced and energy for my muscles. Myoglobin. Myo tells me muscle. This is specifically a protein, hemoglobin, but found in my muscles, myoglobin, and it holds a lot of oxygen. My muscles need a lot of oxygen to contract. And so sarcoplasmic reticulum, what does that sound like? Endoplasmic reticulum, but for muscles, yes. Okay, T-tubules and all of that will come too. Okay, the sarcomere itself is the contractile unit. We said that on a previous slide. It mentions that it has the A bands and I bands. Here we have the terms thick and thin myofilaments. These are things that you're going to have to be familiar with. Thick and thin myofilaments. Thick will be made of the protein myosin. Thin will be made of the protein actin. Myosin is for thick, actin is for thin. Myosin is for thick, actin is for thin. Here is a myofibril up close with the sarcomeres pointed out. This is what you're going to have to be able to label and explain. And once you do it, it's a memorization thing. But I'm going to kind of explain how it all rolls together. I'm going to do it 2 to 3 times. I'm going to see how it goes the first time. One sarcomere is measured from what we call Z-line to Z-line. And this is clearly called a Z-line because it has this zigzag pattern, like a Z. Okay? So this is one sarcomere. Remember, this whole thing is a myofibril that's really long and cylindrical, and it's made up of multiple sarcomeres. Okay? Z-line to Z-line, one sarcomere. Here in blue is our thin filaments. Our thin filaments. They're made of the protein actin. Here in blue are thin filaments made of the protein actin. You can see that the thin filaments are anchored to the Z-line. In the dark red are our thick filaments made of the protein myosin. The theory we're going to learn is called the sliding filament theory, and it's literally these filaments just sliding when your muscles contract. So you'll hear me say thick and thin filaments. Thick again is in red, myosin. Thin is in blue, anchored at the Z-line. The thick filaments are anchored here in the center of what we call the M line, and M stands for the middle. But that area just straight in the middle is also called the H zone. So the M line is within the H zone. Thin filaments, actin, anchored to the Z line on both sides. Thick filaments made of myosin, anchored to the M line. The H zone is that individual middle area. When you say I band, the I band is an area where there's only thin filaments, so it looks lighter. The A band is where there's overlap of thick and thin filaments, so it looks darker. I'm going to go back to a picture real quick just to kind of show you what I'm looking at. Right here, this A band is where thick and thin filaments are overlapping. That's why it's darker. This I band is just where there's only thin filaments. That's why it's lighter. That's what causes these striations, these stripes. Okay, trying to apply it, I'm going to the wrong direction, to what you know already. So again, a sarcomere is Z-line to Z-line. It's composed of thick and thin filaments. The thin filaments are made of the protein actin and anchored on the Z-lines. The thick filaments are made of the protein myosin and anchored on the M line, which is within the H zone. The H zone itself is only thick filaments. You can see that here. The I bands are just thin. A bands are overlap of thick and thin filaments. When this muscle contracts, these two lines come together. And then they go back. And then they contract. And then they go back. And it happens for each sarcomere down the myofibula. Okay? So if you can get it on this level, you study it this way, then it kind of all makes sense as it cascades together. Here is continuing with that chart that was put in at the beginning. It's showing you an up-close look at what the thick filaments look like. I don't like to say, I call these things feedy things. Okay, so I say that the myosin has feedy things, and here the actin has little footholds, or holes for those feet to move into, which is what you and I are going to learn as the cross bridge. Here, this right here, is this taking the muscle mass out. So it's just showing you the line. So you have your thin filaments anchored on the Z line. You have your thick filaments anchored on the M line, H zone, A band, I band. Okay, so it's just pulling out that muscle tissue. Here is an up-close look at a thick and thin filament. Thick here anchored at the M line, the thin on the Z lines. This slide right here puts words to everything I just pointed out. It defines everything. Okay, so if you didn't write down what I said, this is the slide that does that. It defines each thing that I printed out, or whatever. I didn't print anything, but you get what I just said. Anyways. Up close again. This one is high resolution and it's easier for me to look at. Again, it's taking the muscle, the meat of the muscle away. You have your Z lines that are anchoring your thin filaments all the way across. And you can see a protein that will allow for recoil and elasticity. So it'll pull it back and forth. It looks kind of like a spring or a bungee cord. Here are my thick filaments anchored at the M line. Here's my H zone, A band. I band is just thin. Sarcomere is measured Z line to Z line. Questions right now? Just on the anatomy.
What is the H on it again?
H line, H zone,
H zone, yeah.
is just that in the middle where it's just thick.
Okay.
The myosin filament?
Yes, ma'am.
What does that do?
Okay, so the physiology aspect we haven't gotten to yet, but myosin, these little feet are going to grab on to these little footholds, and it's going to be a tug-of-war action. So they're going to grab on, and they're going to pull this way. And when they do that, this comes in. When they're done contracting, the feet will let go, and this will go, whoo.
And it's part of the M line.
What's part of the M line? Myosin is anchored on the M line. Yes, ma'am. Yes, ma'am. Anything else I can clarify?
Do that about 5 more times, probably.
I feel you. I will. It'll end up being like that. And hey, if you just want to hear it over again, just send me an email. And I'll just tell you how much I really like to talk about it. But I'll tell you everything. M line, H zone, I band. I know it's a lot. That's why we're recording it. But I've got you. Please ask questions, and I will be going over this so many times. Hopefully by the time you see it all together, hopefully it makes a little bit more sense. Actin, I already told you, actin is the protein that's found in my thin filaments. Myosin is in my thick filaments. So it's just showing you the name of the protein that is associated with that filament. The myosin filaments are connected at the M line, anchored there, as I mentioned. The actin filaments are anchored at the Z lines, or the Z discs. Okay? So again, this is just putting words to something that I've already said. So the words are here. I just teach it with pictures. I prefer pictures.
Is line and disc interchangeable?
Line and disc are interchangeable. Yes, ma'am. Yes, ma'am. And depending on what textbook you have, it may say Z disc and it may say Z line. So, yes, ma'am. Here's up close. And this is what we're about to study. So, like the anatomy part is Z line, H, all of that. The muscle, the epimysium, myomysium, all of that. I said myomysium, paramysium, sorry. This is the physiology part. So how a muscle contracts, that's what we're about to go into. So here's the physiological part. So anatomy and physiology. And we already know that shape determines function. So the shape of these myosin proteins are designed so that these right here can grab on to these little footholds and pull. When you see this happening in real time, because I understand that this is such an abstract thought, that I wanted to put the videos in here so that you could see them. And specific ones. I didn't want you just to Google randomly and see what you come up with. But when they do grab on, they pull and the muscle shortens. And then when they're done, they let go and it goes back. When we watch the video, it's going to talk about, the first thing it's going to say is calcium. And that's the second introduction to calcium. We'll have another one today. But calcium is needed in every system. Calcium is what starts a muscle contraction. So it'll talk about calcium binding to troponin. Troponin changing shape and allowing for tropomyosin to roll over. And these videos, of course, you can watch over and over again. Then, as a result of that, the myosin heads can then attach and pull. So if there's no calcium, there's no muscle contraction. And if there's too much calcium, there's too much muscle contraction that leads to rigor mortis. And that's actually one of the applications here. So calcium, remember we talked about homeostatic levels. You don't want too little and you don't want too much. Okay? But these will grab onto each other. The sarcoplasmic reticulum, which sounds just like the endoplasmic reticulum, is the area that the calcium is stored. It's holding calcium there. Because in order for me to contract my muscle right now on my big toe, there has to be calcium there already. I can't tell my big toe to contract and then wait for 10 minutes for calcium to show up. Calcium has to be there ready to go. So the sarcoplasmic reticulum is responsible for storing that calcium for me. That's the extent of what you need to know for sarcoplasmic reticulum.
If you have too much calcium, is that what causes muscle spasms?
Sometimes. So calcium is actually a way that you can kill someone. Too much calcium will cause their muscles to go into what we call tetany. And when your diaphragm undergoes tetany, you can't breathe because it's contracted and it won't allow for respiration. And when your heart undergoes tetany, we call it cardiac arrest. So calcium, and actually in the old days, a lot of toxicology tests wouldn't test for calcium. So you could knock somebody off using. Now you know. In fact, if you were to take too much calcium, like calcium supplements, if you took 3, more than likely you would have quick diarrhea or you would throw up within about 15 or 20 minutes because your muscles are telling you that's not going to work. Yeah. So, yeah, too much calcium is a bad thing. Like, you can drink a lot of milk and your body knows how to filter that out because it's not concentrated calcium, so it's a different form. But when you have concentrated, that's lethal. Lethal. Now you know. All right. So here is my myofibril. Look what I just laid on top of it. Sarcoplasmic reticulum. That's it. What's stored in the sarcoplasmic reticulum? Calcium. Because that's what this needs, the sarcomere needs in order to contract. So that's all I want you to know for sarcoplasmic reticulum. Okay? Here's our video. Let me turn on our sound. I'm going to show it to you first, then I'll explain it, and then I'll show it again.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere.
See how they have all come together.
A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat.
See how it's like.
And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum.
When there's no calcium, there's no muscle contraction. So all of this will change back.
Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Fun times. Fun times. So, I wanted to show you
Romeo
that.
and Juliet, Helen and Paris, Tristan and Isolde. These famous star-crossed lovers bring to mind insatiable longing, forbidden love, and tragic separation.
Hey, crash courses are good. I don't know if you've ever watched them. Oh, dang. How did we get so far back? Sorry. That's on me, for sure. Oh. Yeah. Okay, good. All right. So, that overall, it was talking about cross-bridge cycling, and that's what we're going to call our sliding filament theory. Sliding filament theories are where the thick filaments and the thin filaments slide past each other. That's why it's called the sliding filament theory. Okay? That's all that that's referencing. It is initiated. I'm just going to go through key points. It's initiated by calcium. Calcium binds to troponin. Troponin changes the shape of tropomyosin. I don't claim to spell everything right, so I apologize. Myosin. Okay, when that happens, the feedy things on the thick filaments. Yes, thank you. I was like, gosh. The myosin heads will then bind to the actin subunits, or the the footholes. And that will continue. You don't need to know the whole ATP part, okay? ATP is needed for that, but it will continue to do that, and they will continue to move until the calcium leaves. Okay, so I'm going to put muscle or sarcomere contracts. When that contraction is over, calcium exits. Tropomyosin slides back. And troponin goes back to its original shape. I don't know what I'm going to say. Returns to something. To original position. And now the muscle is relaxed. It's no longer contracting. This is initiated and ended by calcium. Towards the end of this, it'll say, what happens if we have not enough calcium? What would happen? Your muscles don't contract. And what happens if you have too much calcium? They stay contracted. Okay? So that's going to be application. Okay? Hypo and hypercalcemia. The sliding filament theory of contraction is discussing the myosin and actin filaments sliding across one another. They form those cross bridges, which is where actin and myosin come together, and it pulls it, kind of like a tug-of-war type scenario. Okay? In a relaxed state, you can see A bands and I bands. They're pretty spaced out. In a contracted state, the A bands and I bands are much closer. Real quick, let me tell you what's happening on these slides. These slides are putting words to the video. Okay? So if I didn't have the video to walk you through step by step, I would go through these words. But it's literally saying the myosin heads bind to actin, the sliding begins. It's putting words to everything. This causes a shortening of the muscle fiber. The Z does come towards the M line. I don't want you to think that I'm negating any of this content. It's just that I showed it to you in the video. Okay? So here, in a relaxed state, you have a solid H zone. You can see where the A band is. You can see a solid large I band. This is what that looks like relaxed. If you're looking at a photomicrograph, here is it contracted. You can see that there's much more overlap. So it's a bigger A band, and the I band is much smaller. So this muscle is contracted. Let's put comparison of the two. So here's relaxed versus contracted. You can see the difference. Okay? Whenever it's contracted, there's much more overlap because the filaments have slided. Thank you. Slid past each other, and they're held onto each other. So this is much more compact. Here, it's relaxed. There's just a little overlap here. When we initially started, and I told you that muscles have to have the ability to come back, they extend, but then they have to have that elasticity. If these two places cannot overlap, that muscle will not come back. So when a muscle's been hyperextended, that means that this part here and this part here are where they can't grab onto each other. Okay, so that causes damage, tears to muscles. All right, now you understand that at a microscopic level. Okay, do you want me to show you that video again? Because I'll be good going on, but if you want to see the video again, okay, we can go on.
I said one.
one more time to see the video. Yes, ma'am. Okay.
And the troponin, it doesn't turn into the trypomyosin. It
No,
just
no,
activates it to make it
right.
turn.
Troponin and tropomyosin are two separate proteins.
Okay.
Yes, ma'am.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
Tell me what you need clarification on.
There's 2 heads
On the
mouse
thick,
heads are,
on the thick filaments, you don't have to know it in that much detail, but there's actually thousands of them. But you saw them paired
Well, yeah,
with
just
2,
pairs,
right?
okay.
Like it's walking like feet, which is why I call them feety things, because they looked like they were like feet, like walking, like as they continue to contract. Yes, sir.
Do we need to know the cycle in order, or just kind of like what happens?
The order is going to be important, which is kind of why I said this here.
Alright.
And on the video, these 2 were flipped. So it's saying that troponin went back to its original shape, allowing tropomyosin to glide over. Ultimately, of course, I want you to know it, that what they're looking for is what causes that to contract, not necessarily the resting part. Okay, so you could learn it either way. This is how I have it memorized, and she clearly said the opposite in that video. I don't know if anybody even noticed that, but she did. She said troponin goes to its original shape, allowing tropomyosin to slide back. So you can do either way. What we really are focused on here is the contraction part, and the contraction ends when calcium exits. Okay, great questions. Additional questions or clarification? Okay, so that was the first technical part I had to teach you today. Bill, let me bring it like that. Second one is the events at a neuromuscular junction. So if I had my way, the way the notes would be centered is I would teach you this part first, because this is what happens second. The nervous system has to tell us to contract before we contract. Okay? So what we learned here is the second part of a muscle contraction. The first part is at the neuromuscular junction. Do you have any idea what neuromuscular might be?
Where your nerves touch.
Literally where your nerve touches your muscle. And we're still on skeletal muscle, so what tells the skeletal muscle what to do? The nerves. Okay? So the nerve telling the muscle what to do comes first, and then the actual contraction occurs. Okay? So the aspect that we're going to look at right now is the nerve telling the muscle what to do. And it uses this term action potential. You and I generate action potentials all the time, and the only way that I can explain this to you without going into the total nervous system chapter is using a cord that you would plug in to a TV. This cord is capable of electricity, and it can generate, it doesn't generate itself electricity, but it will propagate it. It will allow that electricity to travel. That's how your nerves are. If there's no electricity on the end, even though this has the ability to propagate electricity, if there's no electricity at the source, then no electricity will travel. The traveling of electricity is what we call the action potential. So it actually moving electricity through our nerves is what we reference as an action potential. Just like genes can either be on or off, your nerves are either on or off. So we say they're either excited or they're inhibited. And that's going to take us into the nervous chapter. But let's say, we're going to use us right now. After this class, you're mentally fried. I know, because I am too, and I don't even want to teach the next class after this. But some of you go home, and if you're fortunate enough to not have 40 kids that are asking you to do more stuff, you see your couch, and it looks like paradise. So you go sit down, you turn the TV on because you can't find the remote. So you turn the TV on, and then you go sit on the couch, and you're deep couch sitting, and you're like, oh. When it finally comes up, and it's programmed, it's SpongeBob, and you're like, I want to watch SpongeBob. My kids were clearly here last. But remember, I didn't have the remote. So I had to turn it on at the source. In my brain, I want to change the channel, but I have no idea where the remote is. So I don't get up because I don't want to expend energy because I'm wasted. I look around the house, and I see it over there on the fireplace. Okay. I still haven't moved yet. So no action potential has been generated. Even though I've thought about moving, my muscles aren't moving. They're voluntary. I haven't sent them message to move. I'm like, it'll be fine. I can deal with SpongeBob. I don't want to get up. I'm too lazy. So right now, I'm inhibiting. I'm sending inhibitory actions. No action potentials are being generated. But then it's his laugh box episode. And you can tell I have 4 kids because I freaking know all the episodes. But it's like his laugh box, and all he does is laugh. And you're like, how is it legal that they can show this over and over again? And after I watch it a few times, and I was like, okay, I saw the remote, and I get up, and I'm like, oh, no. I get ready to get up, but I'm so tired. You know that feeling. So a full action potential hasn't been generated because I haven't gotten up. It's what we're end up calling a graded potential. I got ready to do it, and I can deal with it, it's fine. And then he keeps laughing. I'm like, oh my gosh. When I finally muster up the electricity to tell my muscles to get up, it generates an action potential. And once I tell myself to get up, my muscles already contract. I can't tell them to stop at that point. Okay, because your muscles, we're going to learn, are all or none. They either contract completely or they don't at all. So while I was sitting there thinking about it, but I wasn't contracting because I was sending more inhibitory signals. I don't want to do it. Even though I want to, I don't want to. So I'm turning them off, I'm inhibiting. When I finally decide I'm going to get up, I send the electrical signal and it goes all the way. It generates a full action potential and I get up and go get that remote. Okay? So that's the easiest way to kind of like parallel it. When that action potential is generated, it goes from my brain, which is telling me to get up, to whatever skeletal muscles need to be activated in order for me to get up. Okay? And in order for you to stand up, it's a full body type thing. Abs, arms, everything. Yes?
Okay. What about like with sleep paralysis?
Oh, yeah. We are going to talk about that.
You tell yourself to move, but you can't.
So individuals with sleep, there's two types. There's sleepwalking and sleep paralysis. Sleep paralysis is to where your muscles have gone into, even though your brain is going, but you're in stage 4 sleep. Stage 4 sleep means that your body is complete, like can't move. Even though your brain is active, your body can't move. People who sleepwalk, there is an actual neurological connection. And people who sleepwalk, that connection has been severed. So even though they're in deep sleep, their body is still active. So they can tell their body what to do. People who are in sleep paralysis, that connection is there, and that's what's keeping them safe from getting up and walking out and doing whatever it is that they do when they sleepwalk. And we actually cover that quite a bit when we go through sleep cycles, and that's in this semester. So that's a really good question. Sleepwalking is a thing, a crazy thing. Anyways, action potential, excitation, contraction, coupling. When you excite it, when you send that nervous system stimuli, that action potential, that's the excitation. The contraction is what we just covered. So the excitation is the electricity, the contraction is the sarcomere contracting. And they have to happen together, which is why it's called coupling. Okay, so this is step 1, this is step 2. Here are all the steps put together in a diagram, and what I think is the logical explanation of how to teach it. Action potential first, and then neuromuscular junction. So your action potential, and I'm going to teach you all these steps, but here is what we just covered here. The action potential goes past the sarcolemma, it releases calcium, it allows the thick and thin filaments to slide past one another, and the myosin heads contract, causing the sarcomere to contract. So this is neuromuscular junction. This is the sarcomere contraction. The cross bridge cycle and the excitation contraction coupling, they're all the same thing. Here is the neuromuscular junction, step by step. And this is what we're going to do now.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron
So here's your
to a
extension
skeletal
cord.
muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction.
Nerve touching muscle.
This junction is a chemical synapse
Technically
consisting
not.
of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so that probably sounded like Spanish. And if you're bilingual, then that doesn't count. But I know that there's a bunch of terms there that you're like, oh my gosh. Trust me. Trust me. Okay, I'm going to get to the picture first because this makes it easier for me to teach it. I have these steps labeled, okay, 1 through 6, and I realize it says 7, but we're going to see the same movement, okay? What we studied right here, this is what we call the motor end plate. Here are the sarcomeres right here. So what we just covered, the excitation, contraction, the cross bridge, that's all right here underneath it, okay? So that you know where we're at. Right here we show that we have yellow, a nerve, coming up and touching a skeletal muscle. And I told you at the beginning, it's a 1-to-1 ratio. So you have one nerve for each skeletal muscle fiber. Okay? So that's how we control them. Terms that were used here. Action potential, which I already mentioned, was the electricity. What this is showing you is the electricity is coming down, and it said it was propagated. When that electricity was propagated, it opened voltage-gated channels. What is voltage-gated going to open and close as a result of? Electricity. So if the electricity is coming, those voltage-gated channels open. If there's no electricity, they stay closed. So these are voltage-gated calcium channels. Calcium, once again, is starting all of this. So here comes the action potential. It opens these voltage-gated channels so that calcium can come in. When calcium comes in, we have these vesicles here, which we've covered that term before, filled with the neurotransmitter acetylcholine. And if you, some of you who have done pharmacological stuff or you've worked with medicines and stuff, acetylcholine is an excitatory neurotransmitter, so it's used over and over again. Acetylcholine is a neurotransmitter in these vesicles that when calcium enters, it stimulates these vesicles to fuse and release that acetylcholine. So normally it stays still, but when you tell it to do something, it releases that acetylcholine so that your muscles react. Here, the nerve is not directly touching that muscle. So we have this area, this moat, if you will, that is called the synaptic cleft, where that acetylcholine kind of floats across. That acetylcholine will bind to integral proteins, that once the acetylcholine is attached, it will open those protein channels. Here's my phospholipid bilayer, my sarcolemma. When it does that, sodium will go in, potassium will go out. And this will cause this next step to occur. Calcium to be released from the sarcoplasmic reticulum.
Is that sodium potassium pump?
This is a sodium-potassium pump. And sodium-potassium pump is a form of active transport, which is why we needed electricity and energy to make it happen. If we don't have the electricity to make it happen, the sodium-potassium pump doesn't happen. What do our muscles not do? They don't contract. Okay, but yes, sir, this is a sodium-potassium pump. All right, when it's done, the acetylcholine will let go of this protein, and as a result, that protein channel will close. That acetylcholine can either be resorbed by the synaptic synaptic terminal. It can be broken down by acetylcholinesterase, and ACE is an enzyme that breaks it down. And so that's actually when I initially taught that to you, I mentioned acetylcholine and acetylcholinesterase, because that's an enzyme that will break it down. And once it's broken down, then it will be recycled. And then the third one was that it can be washed away. In the video it says just 2, it can diffuse away or be broken down by acetylcholinesterase. But acetylcholine could also just be resorbed by that synaptic terminal. I'm going to go through it again and again. But these steps are here listed. Okay? Acetylcholine is abbreviated ACH, just so you know. And acetylcholinesterase is abbreviated ACH ACE. I'm sure it's on here somewhere. Okay, it's obviously not. Yeah, it's spelled out the whole way. But ACH ACE is acetylcholinesterase that breaks down acetylcholine. So, step 1, the action potential is propagated. So we've told that muscle we want to contract. The electricity travels its way down that nerve and opens voltage-gated calcium channels. Those voltage-gated calcium channels will allow calcium to come in to the synaptic terminal, and they will stimulate vesicles full of the neurotransmitter acetylcholine to fuse with that cell membrane and release acetylcholine into the synaptic cleft, that moat-type area. The acetylcholine will bind to these integral proteins and the sarcolemma. These integral proteins, it also mentioned that they were ligand-gated cation channels. Just so you know, ligand-gated is a fancy way of saying chemically gated channels. So if I have a chemical, it'll open up or close in response to it being a chemical. So these ligand-gated channels open up when that acetylcholine binds to it and allows for sodium to come in, potassium to come out, and it will continue to do that, which will then stimulate my sarcomere my sarcoplasmic reticulum to release calcium so that my whole sarcomere can contract. Once this is done, the acetylcholine reversibly binds, so it will let go. This integral protein will close, so this ligand-gated channel closes because there's no chemical to hold it open. The acetylcholine can be resorbed, it can be diffused away, or it can be broken down by acetylcholinesterase, the enzyme that breaks down acetylcholine. The next few slides are going to put all of this in words together. Starts off with calcium, or the electricity. The calcium, voltage-gated calcium channels allow for calcium to come in. Those vesicles full of acetylcholine, that neurotransmitter, fuse. Because calcium has stimulated them, they fuse with that cell membrane, allow for the acetylcholine to be released into the synaptic cleft, where it binds reversibly with these integral proteins that also function as chemically gated channels or ligand-gated channels. When the acetylcholine is there, it opens that protein, allows for sodium to go in, potassium to go out, and that continues. It will actually end up changing the potential, and we'll talk about that later. When it's done, the acetylcholine will let go. That chemically gated channel will close. The acetylcholine can be resorbed, diffuse away, or be broken down by acetylcholinesterase, that enzyme. When this happens, this is the activities at the neuromuscular junction, then the sliding filament theory occurs. If there's no electricity, then none of this happens and the muscle does not contract at all. Notice how both of them start with calcium. The release of acetylcholine is from calcium. The contraction of the thick and thin filaments is because of calcium. Calcium plays a major, major role. I'm going to go back a slide to make sure I mentioned everything. Skeletal muscles. Real quick, the term somatic motor neuron. Somatic means that it's voluntary. Somatic means that it's voluntary. In this case, motor means that it's causing movement. So somatic and voluntary movement. Axons of the motor. Yes. Yes. Neuromuscular junction. One junction per muscle fiber. 1-to-1 ratio. I said that. Okay? Here is where it takes everything that was in the video and right here, and it divides it up into, I think, 4 slides. So it tells you that the axon terminal is stimulated, the acetylcholine enters the synaptic cleft. Here shows you that sodium-potassium pump up close. Sodium going in, potassium going out. This is showing you the let go of that acetylcholine, or the release of that. That chemically gated channel or that ligand gated channel closes. The acetylcholine can be resorbed, diffuse away, or be broken down by acetylcholinesterase. Acetylcholinesterase is responsible for breaking down acetylcholine into acetic acid and choline. You don't need to know that, but that's what that acetylcholine is. This is actually more than 4 slides. Generate of an action potential. Going back to this diagram. Let me show you this video one more time that walks through these steps.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, what questions or what clarification can I offer here? I know. I'm going to just let that fall on you for whenever you want it to fall on you. Thank God it's Thursday. Okay, the terms on this slide. Depolarization, repolarization. At this point, this is where we integrate all of this into a graphical representation. Depolarization, is it going up? Repolarization, is the muscle returning to a resting potential. So whenever it says depolarize, that's that muscle contracting. When it says repolarize, that's that muscle going back to a resting state. Depolarization is going to be associated with sodium ions. Repolarization is going to be associated with potassium ions. So you're going to see that. And it's going to come up. I'm just telling you where we're going to see that. Okay. We're also going to learn now about the membrane potential, because in order for all this to happen, it requires electricity. So right here would be my resting potential. Then we have what we call a threshold. And I'm going to explain all of that. But this is what's going to come up. And because it's electricity, we're going to measure it in volts. Technically, we're going to call it millivolts because we don't want to electrocute you. But we'll get there. Okay, so depolarization. Let's go on this side. And pause it. Depolarization is where the action potential is being generated. And here it's telling you that it's using the sodium channels and they're open. And this allows for that neuron, well, the neuron will stimulate a threshold. If that threshold can be reached, that action potential is generated. Once it's generated, it can no longer be stopped. So remember when I told you were sitting on the couch and you were sitting there and you knew what you wanted to do, but you hadn't told your muscles to do it yet? At that point, because you hadn't moved yet, you were at rest, you had not reached threshold. I told you also that your muscles have two signals. They can either be excitatory or inhibitory. And your muscles actually can calculate. Meaning, if I send 25 excitatory signals and 19 inhibitory signals, my muscle's going to do the excited movement because excited won. If I send 19 excitatory and 25 inhibitory, I'm not going to do anything. Your muscles literally do math with electricity. Seriously, like it's crazy, but it's called summation, and they do it. So when I was sitting here contemplating getting up, I was sending excitatory signals, but I was also sending inhibitory signals. And because I didn't get up, which one did I send more of? Inhibitory. When I finally decided to get up, my excitatory outnumbered my inhibitory. Does that make sense? When your excitatory outnumbers your inhibitory, you reach threshold. Once you hit threshold, and it's going to reference this in your book depending on what book it is, it's like pulling the trigger of a gun. Once you pull it, there's nothing you can do to stop it. So once I hit threshold and I tell my muscles this is what we're going to do, the muscles go straight into the workload. Okay? So this is where my threshold is hit. If the threshold is released, the muscle's going to do work. Once it's initiated, you cannot stop it. Okay? So that's what that's referencing there. Okay, repolarization is associated with the potassium influx, and it goes into what we call a refractory period, depending on what muscle it is. Some muscles have a resting period where they require rest before they work again. Some of your muscles, especially when you're fight or flight, that resting period you can override. Okay, and we'll discuss that as it becomes applicable. Of course, Tyler already mentioned, is this the sodium-potassium pump? And it's called the sodium-potassium pump because sodium and potassium are being pumped into and out of that muscle fiber. Okay? This is showing you that depolarization versus repolarization. When that cell is depolarized, the channels for sodium are open. When it's repolarized, the potassium channels are open, the sodium is closed. So depolarization is associated with sodium. Repolarization is associated with potassium. They technically they don't, but they do for what we show here. They both can be happening at the same time. That's why yes, they can happen at the same time. So we have these integral proteins. That's a biological concept. I'm not going to go into it right now. But technically, they have a specific shape because they have a specific. Yeah.
Potassium moves in.
Yeah, okay. So potassium leaves, sodium comes in. Yes. Okay. Are you good with that?
So, okay.
No. Go ahead.
Sodium moves in when it's depolarized.
Mm-hmm.
Right? Potassium moves out.
When it's repolarized. So, did you say potassium moves out?
Yes. Potassium moves out when repolarized.
Yes. Okay, good deal. So here I have what I attempted to draw here. I mean, I drew it, but it looks cooler here. What this is showing you, it gives you a number right here. It says negative 95. This is referring to the voltage, and we're going to measure it in millivolts. Negative 95 millivolts, let's say, is resting potential for one group of your muscles. I will tell you that as we get through this, muscles have a different resting potential. The muscle that's easily stimulated might have a resting potential that's really close to threshold. Like your eye muscles, your extraocular muscles, you move those really quick, and so they can't be really rested. They're kind of stay there in tandem all the time waiting for you to move. Okay? But large muscles in your legs and abdomen and your arms, they have a lower resting potential because it takes more work to get them to move. All right? So anyways, this is showing you a textbook number, negative 95 millivolts. We're just using this generally speaking. This is not for every single muscle. There would be a threshold technically that was supposed to be here. Your threshold number is negative 55 millivolts. That's the textbook number. So when it goes from 95 to 55, that's when this, and I'll get there, but that's when this all happens. Depolarization is when the muscle's contracting. You can see that it's associated with sodium. Here it hits its peak, which is around positive 30 millivolts, and then it starts to go back down to its resting potential. Repolarization, potassium is exiting. Okay, so it is what I drew right here. It just looks neater. All right. The latent period, that's what this next section is going to reference. Let me show you here. Oh. And I'll come back to, never mind, I thought latent period was going to be next. We have a refractory period. Latent is right here when it's kind of waiting. Then it contracts. Then you have absolute refractory and relative refractory. During this time, as the muscle's coming down to relax, it won't let you do anything else. It says, I have to relax. Relative refractory says, if you give me enough of what I want, I'll do what you want me to. But otherwise, not. And we're going to come to those terms. I just wanted to reference them really quickly while I had this diagram. And it's mentioned here, the latent period. It's the time between those events. So that kind of resting period, that wait period. Okay? That's when that calcium is going in and out, is what actually stimulates the latent period. Here, this right here is showing you what it causes. This is that neuromuscular junction in order for the sarcoplasmic reticulum to release that calcium. So this is all of those steps again in words, and then ending with the sarcoplasmic reticulum releasing calcium so that the muscle, the sarcomere itself, can contract. So this is just putting that all back into words. Rigor mortis is where, well, hopefully everybody's kind of familiar with what rigor mortis is. So your muscles are stuck. The reason your muscles are stuck is because those cross bridges never let go. The cross bridges never let go because the blood flow stopped. The blood flow stopped because you died. Okay? When the blood flow stops, the calcium movement stops. When the calcium movement stops, the muscle stays where it is. ATP, which is that energy, is required for your muscles to relax. And because you're dead and there's no oxygen, you're not making any more ATP. So we can measure the time of death based on how far into rigor mortis you are, how contracted your muscles are. In the event that you find a body that's been in the same crazy position for 24, 48 hours, when you pick that body up, it doesn't just go back to its regular position, its anatomical position. If you find somebody who died in a crouched position or they were in a small setting, when you pick up their body, it comes like that. Like it stays all together. And the ways to get those muscles to relax so that you can actually study them, you can tear them, you can cut them, or you can pump them full of like a saline solution or a preservative to try to get those muscles to get flow, to get that calcium to release. It's not an easy task, but it's doable. So sometimes when you have bodies that, they're in the morgue and they kind of do those like flexing type things, a lot of times those bodies were in a different position when they were initially found, and they pumped them with the fluids or whatever, and those fluids diffused away, and so they came back to that position. And you're like, oh my God, they're moving. Not really. It's just those muscles reacting. So it's one of those explainable things. Okay, principles of muscle mechanics, muscle tension. 2 types of muscle tension, isometric, isotonic. Big difference. Isometric means that your muscles can't shorten because whatever it is too heavy. Isotonic means that you can lift it. So the way I remember it is isometric is you can't lift it, it's really heavy, and isotonic, you can lift it. It uses the term force and load. The force is you, the load is what you're trying to lift. So for isotonic, my muscle force exceeds the load of this cup, so I can pick this up. As a result, my muscle shortens, and I can move this cup because the force of my muscle exceeds the load. This is isotonic. Isometric says that I try to lift something that's heavier, the load exceeds the force that I can put forth. So even though I'm trying to lift it and my muscle fibers are working, this is too heavy for me. So I'm flexing, but my muscles aren't shortening because the load exceeds the force. Okay? And you're going to see those 2 terms here in diagrams. So this is isotonic. This is something that I can lift, so my muscle shortens. An isotonic contraction. Isometric contractions. I pick it up and I'm contracting, but it's too heavy for me. So even though I'm trying, my muscle doesn't shorten. I don't move. Okay, so I'm still exerting the force. It's just not enough. Questions on the difference between isometric and isotonic? A motor unit. This is how our muscles prevent us from getting tired, and the term fatigue is used here. Our muscle fibers divide themselves up into motor units, and we'll say, for example, 3 motor units. Okay? So I normally, in a shorter class, I would have us all get numbers. We would go 1 to 3, and I would say, okay, if you're a 1, stand up. So you would stand up because you're holding that weight first. But the thing is that, let's say it's this cup. And some of you have done this before, like you're holding a drink and you hold it for so long your hand gets tired. Like it's uncomfortable. Some of you hold your phone and you video and your pinky gets tired from resting it on there. This is the principle that's taking place right now. So I would say 1, stand up. And right now, the 1s, because this isn't really heavy, I just have a small number of motor units working. The 1s stand up. As the 1s get tired, I don't drop my cup. The 2s take over. So I'll ask the 1s to sit down and the 2s stand up. So right now the 1s are resting while the 2s are doing the work. As the 2s begin to fatigue, I'll ask the 3s to stand up, and your muscles do that. That's why you can hold things for a long time. Light things, not heavy things. It's because those motor units will cycle through, and they kind of help each other out. And that's only on light loads, of course. When it's a heavy load, we have a different situation. So we're going to end up, and I'm going to show you a diagram here in a minute after I go over the twitches and the summation that shows you all of that happening together. A muscle twitch is nothing more than a single muscle contraction. People are saying, well, you know how sometimes you can feel it twitching and you feel like everybody sees your whole face shaking, but it's actually just you who knows it's happening because you can feel the muscle contracting. But because it's only happening in one little muscle fiber, nobody else can tell but you. A muscle twitch is just one contraction. It doesn't necessarily have to be lifting weight. There doesn't have to be a force. It can just be a random muscle twitch. Okay? It doesn't really move anything. It's a muscle twitch. Here's kind of what we're looking at. This is not a muscle twitch, but this is showing you the latent period, and here is showing you the period of relaxation. So this is depolarization, repolarization. Even in a muscle twitch, you would have depolarization and repolarization. It would just happen super quick. Okay? But nonetheless, so the period of relaxation. Here is showing you different muscles in your body and the time frame that we can talk about that they contract. The smaller the muscle, the quicker they are to contract, the quicker they are to relax. So here it says extraocular muscle, and lateral rectus. So these are the muscles on the sides of your eyes that move your eyes to the side. You can see that they contract really fast and they relax really fast because this is a smaller muscle. As you get to bigger muscles, we have a slower contraction and a slower relaxation. What's the gastrocnemius? The calf muscle. Okay, so the calf muscle has a shorter or a longer period. Then we have the soleus muscle. What's the soleus muscle? It's still in your thighs, yes. Your leg. I said thighs, but legs. Okay, graded muscle responses. So those are just diagrams referencing those two. Graded muscle responses are when I was talking about excitatory versus inhibitory. A graded response means that you are not reaching action potential. You try, but you don't do it. You try, but you don't do it. So a graded response was me sitting on that couch, and I would start to try to, no. Graded says that I get close to threshold, but I don't. How do I know I don't reach threshold? I didn't start moving. Once you hit threshold, you immediately get going. Graded says, this is you like, no. Once I decide to full get up, I've reached threshold and an action potential is generated. Okay? So graded responses, they're going to say that they could technically be depolarization or hyperpolarization, and we haven't covered hyper yet. We're going to get there in just a moment. So graded means that it did not reach the action potential. Here again talks about a muscle twitch being a single muscle contraction.
Question on the muscle twitch.
Go ahead, right here.
Wouldn't that be involuntary?
No, because you're technically telling it to happen. It could be reflexive, though, which means that your brain, it's not like when we get to the nervous system, we'll talk about the fact that some things go to your spinal cord and some things go to your brain. And when it goes to your brain, you actually integrate it and you try to figure out, what am I going to do with this information? When it goes to your spinal cord, it's just there's nervous system integration, but it's a lot less higher level of thinking. So it could be spinal cord.
It's like pre-programmed.
It is. Something that's happened before, something that you've experienced that your body, so there could be, and I'm pointing to my eye because everybody talks about their eye twitching, but it could be that your caffeine levels are really high. So caffeine tells your nervous system to be stimulated. So this is just a muscle that's manifesting that stimulation as a result of that. Does that make sense? Because caffeine is a neurotransmitter that would work like that. Here, you can see we have a single twitch. So This is not necessarily saying that this is threshold, so I don't want you to coincide that and this, or correlate those two. This is saying I just have a single muscle contraction and relaxation. I just have a muscle twitch. That's it. Just a single muscle contraction. Wave or temporal summation, also called incomplete tetanus. This is when you lift something that's kind of heavy, and you have to continue to tell yourself to hold it. Even though you're not telling yourself hold, 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 you really are telling yourself hold, hold, hold. When you see people do that textbook thing where they make you hold up a textbook or something that's pretty, like even this laptop would work for this, that they say hold it out in front of you as long as you can. I can hold it out here, but after a while, my muscles are going to start shaking. Because right now they're alternating motor units. 1's working, 2's working, 3's working. After a few minutes, my muscles are going to fatigue. And they're going to tell me, stop. But I can keep sending the signal that says what? Hold it, hold it, hold it, hold it, hold it, hold it, hold it. So I'm continuing to send action potentials that are telling me to hold this. So this is what this looks like. I had increased that muscle contraction, and then I'm telling it, hold it, hold it, hold it. When I set it down, everything goes back to relaxing. So you're continuously sending the stimulus to hold, 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 hold. Okay? As I lifted it up, this is it going kind of up like that. And then I'm telling it to hold, 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 hold. Okay? This is the majority of your muscle contractions. Your muscle contractions, unless, of course, they're a twitch, this is kind of what they look like. Okay? We call this wave summation or incomplete tetanus. So tetanus would be a full contraction. So this is incomplete tetanus. All right? This is a load that I can pick up. This is not a super heavy, heavy, heavy load that I can't pick up. This is a load that I can handle, and this is what that contraction would look like. The opposite would be tetanus. Not opposite, but the extreme would be tetanus. And just to be perfectly honest with you, it is really difficult to put yourself repeatedly into a complete tetanus. Okay, complete tetanus means a complete muscle contraction, usually of a force more than you can handle. Usually a load more than you can handle, not a force. A load more than you can handle. You see a lot of references to this when people are using really heavy weights, or when they're talking about that superpower you get whenever like adrenaline takes over and you can lift up a car, or you can do something like that car. I mean, some people could work out lifting cars. I get that, okay? But like, you're not going to see me trying to lift a car over and over again. But when that adrenaline rushes, people talk about how you can go over and you can lift up something where the load normally exceeds, but you do it. That's a complete muscle. Like my muscles are completely contracted. It looks like this. And how fast do I fatigue? But I'm trying to lift a car. I'm fast. Okay, I'm 140 pounds and I'm trying to lift this car. I'm going to do it, but whoever's in that car better squiggle out really quick, because I only have so much that I can hold. Does that make sense? So complete tetanus is not a normal occurrence in your everyday life, unless you're lifting super heavy weights, like we could see bodybuilders and stuff like that doing that. Yeah. Okay, but this is not normally something that you would undergo. Whenever they talk about tetanus, like your tetanus shot, whenever you have a tetanus infection, which is technically caused by a bacteria, it causes complete contraction. And that's why it's called tetanus. And when you have complete contraction of your heart, tell me what happens to you. You die. Yeah. So when you talk about your tetanus shot, like you step on an iron nail or something metal, and they're like, oh my gosh, have you gotten your tetanus shot? That could have a bacteria that could cause the infection, that could cause complete muscle contraction, which would be called tetanus.
Why is it just metal?
It just happens to be that it doesn't necessarily have to be metal, it just that bacteria is more prevalent once you have an infection when you step on dirty metal, like iron usually as well. Yes. Good question. Good question. Okay. Recruitment principle. And I feel like once I explain this that it kind of makes sense. Your muscles don't want to work if they do not have to. And most of us are the same way. Okay? So if I have to do something and it's lifting a light load, I want my small muscles to do the work first. And my small muscles are like, I've got it, not a big deal. My big muscles don't want to move because that requires more ATP which requires more energy reserves from me. Okay? So my small muscles will try to do it first. If my small muscles can't do it, they'll say, hey, I need some help, and I have intermediate-sized muscles that come in and try to work. If those two can't do it, then the big dogs come in. But we always try to refrain using the big muscles because they require more ATP, more energy. Okay? And I'm going to show you what that kind of looks like here in a moment. A sub-threshold stimuli. Sub-threshold means that I don't reach threshold. So what happens? Nothing. Sub-threshold means I don't hit threshold, nothing happens. A threshold stimulus says I hit threshold, so I generate an action potential. A maximal stimulus means that's the maximum amount of stimuli that that muscle can handle. Even if you stimulate it more, it can't contract anymore. Okay? And that it depends on what you're looking at and what muscles we're referencing. Here is a great example of motor units. So each of these would be like me numbering you 1, 2, 3, and then I say the 1 stand up, the 2 stand up, the 3 stand up. If I'm lifting something that's pretty light, the 1s are like, okay, I'm good. Here, they could even rotate 1s, 2s, 3s. Here, it's saying this is a little bit more work than we thought. We should probably recruit larger muscles. When I'm here, my big muscles are doing the work. So your motor units, they'll continue to rotate until you've reached maximal tension, maximal force, and then all of them are engaged. When I'm right here, I can do this for a long time. When I'm right here, I can't do that for a long time. This requires a whole lot of ATP all at once. So quick force, quick fatigue. Slow force, slow fatigue. Recruitment works on size principle. We go from smallest to medium to large. And here's this. So here the small ones, they tend to do the work first. They're like, like you would do if you're an oldest sibling, like I am. If my parents tell me to do the dishes, I'm going to say, okay, I'll get the dishes done. And then as soon as they leave, I'm going to go get my sister. And I'm going to tell her she has to do the dishes. And if she fights me, I'm going to fight her back, and then she's going to do the dishes. Right? Like you tell the younger ones to do it. Because you're the big dog. You're the first. You're the oldest. Okay? You're not trying to do any extra work. So the smaller ones do it. The middle child, I'm sorry if you're a middle child. I'm sure you've been back and forth your whole life. And then the only when things get crazy out of hand does the oldest child step in and take care of it. And that's the same principle we see with our muscles. Small, medium, and then large. Muscle tone. Your muscle tone is due to the amount of activity that you undergo. So if you work your muscles are more toned. And if you don't work your muscles a lot, then they're not so toned. Your muscle tone is also a reflection on the amount of adipose that you have, body fat percentage. So you could have pretty toned muscle, but you might also have a high body fat percentage. So if you have a high body fat percentage, it's really difficult to see those toned muscles. How do you tone your muscles? You do physical activity. And we tend to put the word exercise on it, but you could do air conditioning work, and you're picking up air conditioners all the time. You're not in a weight room lifting, but you're still lifting weights. You're still doing physical activity. And the more you use a muscle, the more likely it is to hypertrophy, which means to get bigger, so it becomes more toned. And you want to keep your muscles firm, healthy, and ready to respond. So you do this by taking care of yourself, drinking water, and doing physical activity. I think I've already talked to you about ATP. Have I? Please say I have. Good, thank God. So if I decided to go run today, the ATP I had would be used up. We talked about this, yes? Good. So the ATP that I have ready to use right now will last me a few seconds. If I use all of it, if I stop breathing, I will not make near as much ATP. I'll make ATP for a few more minutes, and then I'll stop. Of course, then I'll die. And then it'll start the rigor mortis process. But what I have right now, I could use in a few seconds. The fact that you breathe means that you can continue to produce ATP. So whenever you're working out, especially if you're doing endurance workouts, and say you're being trained by someone, what do they tell you need to continue to do? Breathe. And how do they tell you to breathe? In through your nose, out through your mouth, so that you're humidifying that air and moistening it before it hits your lungs so that it's working at its maximum potential. If you've ever tried to work in the cold or run in the cold or work out in the cold, and your nose is running and you're breathing through your mouth, it becomes painful to your lungs when that air is so cold. Okay? So there's a reason why there's that pathway there. Here it talks about ATP being generated by creatine phosphate. And I like to address this especially because there's a huge misconception. Like when you go to the nutrition store and you buy creatine powder, and you're like, oh, this is going to make my muscles big. And I'll get you when we go to our break. When you get that creatine powder, it tells you, like you have this idea that, oh, it's going to make my muscles big. Creatine powder does not make your muscles big. Creatine powder increases the rate at which you generate ATP. So it gives you kind of a higher boost of ATP, which gives you more energy to work out. So if you have more energy to work out and you use that energy to work out, what happens to your muscles? They get bigger. Okay? But if you drink that creatine phosphate or you consume it and you don't use it, then it's not doing you any good. You're literally just spending money to do nothing. So creatine phosphate helps you regenerate ATP. In fact, we call that phosphorylation. In the event that you stop breathing, you hold your breath like you're lifting weights or you're sprinting. You don't take meaningful breaths while you do that. We call that anaerobic activity. That's why you can only do it for a very short time. As soon as you do that, when you're finished with it, tell me what the very first thing you do. You just sprinted. At the very end of that sprint, what's the very first thing you do? Because you're trying to get that oxygen back to the levels it was before you exercised. Okay, so anaerobic produces lactic acid. When you do a lot of anaerobic activities like lifting weights or sprinting, it causes soreness because lactic acid builds up. You're supposed to make sure you're drinking a lot of water and stretching in order to get rid of that. So this is talking about the cellular respiration process, which you don't have to be familiar with here. But this is how you actually create ATP. Whenever you are not breathing, though, it will leave lactic acid in your muscles. And when you talk about somebody being sore after they work out, it's because of lactic acid buildup. The only way to get rid of that soreness is to use that muscle again. So you have to do activity again. Okay? You're going to have to do some type of activity. Some people say, oh, I need to rest. I just really overdid it yesterday. No, you need to get up and do something. Okay? Especially if you want to increase the size of your muscles. So here it talks about lactic acid and that breakdown. Aerobic pathways, which is what you and I go through all the time. When you're aerobically working out, like doing something like running or swimming, swimming is actually anaerobic, but you're doing something that you have adequate access to oxygen, you're less likely to build up lactic acid, so you don't become sore after doing that. So that's what the difference is in aerobic versus anaerobic. Aerobic, you can produce a whole lot of ATP. Anaerobic, you don't. You produce a whole lot less ATP. So that tells you the difference here. Anaerobic is no oxygen access. Aerobic is oxygen access. Again, anaerobic activities that you may do and still live, like hold your breath, you're lifting weights, you're sprinting, you're swimming, that's also anaerobic. Here is you doing a short duration exercise, like a sprint. You start off really hard, okay, and at the end, you have that need for oxygen, so you're gasping. Here, prolonged, and it's showing like endurance activities, you can run at a steady rate where your aerobic levels, you're respirating well for a long time. Post-exercise oxygen consumption. That's a fancy way of saying when you're done exercising, the first thing you want to do is breathe. Yeah, take a deep breath. That's all that's saying right there. Heat production. Whenever you're working out, what do your muscles release? Hopefully, this is what happens. When you're working out, you get hot and you sweat, like you're releasing heat. So your muscles are doing work. So you're sweating. That's hopefully what happens. Hopefully you're not working out and not sweating, or not working your muscles, because then you're really not. Anyways, dangerous heat levels can cause damage. I think that we are familiar with that already. Whenever you're really cold, you shiver and your muscles contract in order to produce heat. The force of a muscle contraction depends on a few things. So it could be the number of muscle fibers, the size, whether I'm using small, medium, or large, how frequently I'm telling them to contract, and how far that muscle is stretching. The amount of muscle stress could also increase that force. This right here is this diagram. This is the words for it. So it's using the arrows, like increase this, decrease that. That's all of this right here. So an increase in the contractile force of a muscle, it could be affected by the number of muscle fibers recruited, the size, the frequency of the stimulation, or how far the resting length is. So is it overlapped like this, or is it here? Like, where is it? What am I grabbing onto? Slow and fast fibers. We don't have to know these in detail, but slow fibers, they contract slowly and they relax slowly. Fast fibers contract fast and relax fast. That can be a problem, but the amount of ATP needed for slow fibers is lower, and for fast fibers it increases. When you have a heavy load, you can't hold it very long. So that's what this is showing you. The time and the amount that the muscle shortens. When the load is heavy, I can't lift it very high, and I can't lift it for a long time. In an intermediate size load, I can lift it higher and I can hold it for a little bit longer. In a light load, I can lift it and I can hold it. Here is telling you the velocity of the shortening. How fast can I shorten my muscles in relationship to the load? When there's no load, I can shorten my muscles very quickly. When it's a heavy load, I can't really shorten my muscles quickly at all. Okay, so that's what this is showing you here. In aerobic endurance, what this is basically saying, and I had covered this when we talked about ATP, if you start working out today, you're going to get tired. But after you do it for a certain extended period of time, your body becomes accustomed to it because you actually create more mitochondria, you create more blood vessels, which increases blood flow and the amount of ATP that you produce. So that's what this is talking about right here, is referencing endurance activities. What happens whenever you do anaerobic activities? It increases muscle hypertrophy. This is the fancy way of saying your muscle fibers get larger. So your muscles get bigger when you do things like sprint or lift weights. Not aerobic activity, but you do anaerobic activity, and that causes your muscle fibers to increase in size. Again, the more you do anaerobic, the more mitochondria you will eventually produce, and the more blood vessels that you will have, more capillaries, so that you can keep oxygen to those mitochondria. We increase the size of that muscle fiber, we increase the strength of that muscle holistically. Overload principle, basically it says, if you overload your muscles fall out on you. So there's people who work out, and eventually they just get tired of working out, their body gives up. Okay, now you do want to work your muscles more than like an easy load. You want to do a medium-sized load to a heavy load in order to cause that muscle to increase in size, that muscle fiber. Overuse means that you're working out that same muscle group over and over again and you're not giving it a chance to rest. The best workout activities alternate anaerobic and aerobic activities so that you're taking care of your muscles, increasing blood flow and your endurance, as well as your muscle tone. What happens if you don't use a muscle? You lose it, so that's what that means. Disuse leads to atrophy. That could be the result of you being paralyzed, or perhaps you broke a limb and you're not using that muscle anymore. Without neural stimulation, so for example, people who become paralyzed, say I'm 37 years old and I become paralyzed. My muscles have a certain shape right now. When my brain no longer tells my muscles to move, they start to decrease in size. So I'll start off with legs the size of what I have now, but over a period of time, my legs will go from where they are to much smaller. So a lot of times when you see people who've been wheelchair bound for a very long time, their lower limbs are very, very thin. In fact, their muscles are pretty meat. And of course, they could not support their body weight. Once you get too far, there's nothing that you can do to rehabilitate those muscles. So if you do get an injury or an accident, then you're trying to start physical therapy in a reasonable time frame so that you can regain reuse of those potentially. The longer you go without using them, the less likely you are to regain use. That is it for skeletal muscles. Here's smooth and cardiac really quick. Smooth muscle is found in your organs. You have two layers. You have a longitudinal layer and a circular layer. They lie perpendicular to one another so that when one contracts, then the other is at rest. So they do this push-squeeze action. Push-squeeze action. We have that. We end up calling that peristalsis. You can see here that the fibers go this way here, and on this one, they're longitudinal. So they go in two different directions. Because they do that push-squeeze, they can move things through either the esophagus or the intestines, wherever it is that they're moving through, because they have that asynchronous contraction. So we have a longitudinal layer, a circular layer, so that's what it's mentioning there. This allows for peristalsis. Peristalsis is that movement, those contractions that move food, chyme, all of that through your systems. Here's that same diagram from earlier. Again, neuromuscular junctions and smooth muscle. There's no neuromuscular junction and smooth muscle. It's not a 1-to-1 ratio. They have what we call varicosities. Varicosities look like, I think they look like those netted Christmas lights. And you kind of just lay them across all that muscle. So like this right here could impact this range right here. We call it a field, a working field. So each one kind of has its zone where it does work. And if you look at them up close, they have all the organelles. This is clearly nervous tissue, it's yellow, but it's got the mitochondria and all the various organelles that will allow for that muscle contraction. So this will release its neurotransmitter and it will affect that area. It's not a 1-to-1 ratio. It could be like 1 to 10 or 1 to 20, depending on where it's at. That's also why it's very difficult for people to pinpoint abdominal pain. When they know their abdominal hurts, but they can't tell you specifically where. I can tell you when my bicep hurts, because I know it's right there. That's why it's very difficult to pinpoint this. And viscera. Contraction of smooth muscle. Remember that this is involuntary, and it's a very slow contraction. So when you're digesting food, it's not like, it's very slow, and it helps to mix, and it's a process. You don't digest food in 30 minutes. It takes hours depending on what you're eating. And when we talk about the heart, we'll have pacemaker cells. We do have them in smooth muscle as well, but more so in cardiac muscle. The process of a smooth muscle contraction, it's initiated by calcium. That's all you need to know there. It does also carry out the sliding filament theory, but that's all you'll need to be familiar with there. Same diagrams from earlier. Regulation of contraction. Your smooth muscle contracts when that nervous system tells it to contract. Calcium levels get high. And it talks about local chemical changes. If something's happening and it needs to respond, it will. You don't need to know the details of all of that right now. And I know that your brains are fried. Hyperplasia. This is specific to smooth muscle. So skeletal muscle we can grow by working it out. Hyperplasia is growth of smooth muscle. As you grow naturally, your smooth muscle does increase proportionately to you. But after you hit an adult stage, your smooth muscle shouldn't really grow anymore. Unless you're a woman and you become pregnant. Your uterus will grow in size as the baby develops. So that muscle has to get bigger. But also, when there's no baby there anymore, guess what the uterus does? It gets smaller. Okay? So hyperplasia is just the growth of smooth muscle. Like I said, it happens naturally with your growth, but then it also happens with women during pregnancy. Continuation of that chart again. Types of smooth muscle. Smooth muscle, it's smooth muscle whether it's in your blood vessels, your organs, your lungs, wherever it is. As far as how it contracts, that's unique to that area. And we'll talk about those as we get to those different systems in those different areas. Developmental aspects of muscles. You're born with muscles, but you can't control your muscles. The involuntary ones, they're clearly being controlled involuntarily, but your skeletal muscles, you mature in your control for skeletal muscles from head to toe. Your nerves have to be neurolated, and we're going to cover that next week. But once those nerves start to grow, that baby, the first thing they can do is lift their head, and we talked about this when we talked about bones. So as those nerves begin to mature, they can lift their head. Then it takes a baby much longer to be able to walk because those nerves have to mature and be myelinated all the way down to their toes. So that could take 10 months. That could take 18 months. That could take 2 years. And there also has to be a motivational factor there. If you carry the baby all the time, they don't really have a need to walk. So that could be a thing too. That's also why the last muscle that most children can control is their urinary orifice. And so sometimes boys will take longer to potty train than girls because that nerve hasn't been myelinated yet. So, and that's unfortunate because it is a crutch for a lot of abuse cases. But it takes a child longer to potty train the longer the time it takes for that neuron to myelinate. Because you actually have to control that orifice. Like you can tell yourself, even if your body says it's time to pee, you can tell yourself, this isn't a good time. And you hold it shut. Like you're sending that message, nope, not peeing, not peeing, not peeing. Okay? So this is the way that you develop, and so on. Smooth muscle, you can continue to create that throughout life. Skeletal muscles, once you make it, it's pretty set. Cardiac muscle, once you make it, it's already set. Muscular development, I already said that, neuromuscular coordination. Your neuromuscular coordination should peak around your adolescent years, 18, 19, and you can maintain that if you continue to work yourself out, but if you don't, then usually we see muscle coordination decreasing with age. Females tend to have about 36% of their body is muscle, males a little bit more. That's natural, but are there females that are bigger than males? Yes, so these are textbook numbers, just so you know you can't just apply that to everyone. Okay, sarcopenia. Penia, anything means low amounts, so sarco means low muscle amounts. So as we age, we tend to see muscles decrease in their size, which means we decrease in strength. Okay, it also mentions here that atherosclerosis, so a buildup of anything really in the vessels, could occlude blood from getting to different muscles. We call that intermittent claudication, and that could keep that muscle from actually doing work. It could damage it, actually scar it. It depends. There's a lot of different ways to do that. I reference that. Muscular dystrophy. Muscular dystrophy. So muscular, and what's happening to my muscles? They're breaking down. I'm breaking down the muscle. So the muscles will enlarge, and then I'll have a bunch of scar tissue. So it says connective tissue deposits or fat deposits. This causes the muscle fibers to atrophy. And when they atrophy, those muscles get smaller, and I can no longer use them. Duchenne's muscular dystrophy is a genetic condition, and a lot of individuals who are not treated will die early in life because their diaphragmatic muscle gives out on them, so they suffocate. There is no cure for muscular dystrophy, but I will tell you right now, especially if you know somebody with muscular dystrophy, there's quite a few drugs now that can prolong the life of that individual. The thing is to figure out if it's genetic or if some type of mutation occurred. If it's genetic and your kids are going to get it, you don't want to have kids that are going to have it because you don't want them to suffer, but so on. Okay, holy balls.